It's my pleasure to introduce Edric Choi. Edric is a PhD researcher at the Julius Lux Lab at Northwestern University. He's part of the Center of Synthetic Biology and National Institute for Theory and Mathematics in Biology. He studies RNA structure and dynamics using high throughput chemical probing. He's done some really cool work in this area, and he's here to share this with us. Yeah, over to you, Edric. Really
All
looking
right.
forward to this talk.
Great. Thank you. Yeah, thank you, Chaitanya. I think I was very happy to get the invitation as well. Because I saw the past speakers and they were all kind of my heroes, like it's the people I read papers of all the time and inspired a lot of the work that you're seeing here. And so yeah, I think I'm very excited to share this work. We recently put this out as a preprint, so this is primarily like that paper. And so if you don't want to read it, you can just listen to me and ask me questions. It's the same title, the kind of QR code is there as well. So yeah, I'll dig in. So the title is Ubiquitous Low Energy RNA Fluctuations and Energetic Coupling Measured by Chemical Probing. So we all know that RNA structure is important, and I'm really glad I don't have to belabor the point with this group. But we got to think a little further and ask, what is it that gives us those structures, right? And the answer really is energetics, thermodynamics, right? We might be more familiar with kind of nearest neighbor parameters that allowed us to predict structures. But obviously for design and other things like that, knowing kind of which structures are more likely, less likely, what the energy landscape looks like, those are all kind of very, very, very important things that's underpinning kind of structure, right? And so that's what this talk is going to be about, is actually a lot of energetics, and you'll hopefully see the connection with chemical probing as I go through the slides. So what's surprising to me when I was reading through all the literature thinking about energetics is that we don't actually know with certainty what the energy of opening a base pair is. And that's surprising because I guess we take these things for granted and we just use tools and yeah, we know kind of base pair should form or not form, but the numbers beneath it is still debated. So the earliest numbers came from the 60s or 70s. Walter Englander did some tritium exchange experiments. And these energies suggested, this is for nucleic acids, is in the range of 1 to 3. By the 80s, NMR became a thing and they shifted over to immunoprotein exchange. And now all of a sudden they're saying it's actually 4 to 10, right? And so it's much, much bigger than that. And this was in the 80s, and there's like apparently a huge field of people studying just base pair opening and energies, and I'm not an expert in that. If you're one of those, I'd love to talk to you more. But the main idea is that we don't actually know what these energy numbers are. It's still being debated. So the most recent one came from a single author, article from Walter Englander just earlier this year, January, where he again fought for this smaller value from his earliest tritium exchange. And he had this fun theory of, it's called the soliton theory, where maybe like ranges of 10 base pairs open at the same time. So stacking stays intact, so the energy is lower. So you'll see that doesn't really apply to our case because our stuff are really small. But you know, really the point is it's still hotly debated and we don't actually know. Why does this matter, right? Biologically, you might say like, okay, well, who cares about 2 kilocals or 3 kilocals versus 5? It matters because percent population, when you do the math, you do the exponents and stuff, it results in a huge disparity in population, right? 7 kilocal would be like 0.0008 and 2 kilocal would be around 3, right? And that's huge for things, anything, right? So like protein binding, RNA binding, ASO binding, designing small molecule stuff. These are huge orders of magnitude difference in accessibility, right? This is base pair opening. But at the end of the day, we still don't really have a clear definition. And it's tricky because access to these things are still limited, right? So we have obviously nearest neighbor, and that served us really well for predicting these delta G MFE. So that's kind of like you sum the entire thing and you say that's the minimum one and that's your most likely structure. And they're very accurate, but they're largely limited to canonical base pairs. They're not context specific. again, nearest neighbor means literally only the nearest neighbor matters, right? And so beyond that, it's kind of just looking at their nearest neighbor and so on. And so it's not the most generalizable, and it fails in more complex RNAs. And then next we have this M simulation, which is the opposite of this, where you have high perfect resolution of every single atom. You get kinetic information, but the accuracy depends on the force field, right? And so if you want to say, I want to know what this looks like in cells or even in a particular salt, right? That all affects or is dependent on your force field. It's going to be computationally more expensive. There are coarse graining and stuff like that. But at the end of the day, it's still very difficult to define what is open state, right? In MD, it's almost like there's too many, I guess, like you have such resolution that you could see all of it. And so then it's up to you to call what's an open state and what's not. And we go back to the same problem earlier where the experiments are kind of in disagreement with each other and there's no clear answer. So my favorite of these three is NMR. So obviously with NMR, you also get atomic resolution. It's an actual experiment in solution. You get kinetics. You can get it across large timescales, broad timescales, all sorts of different things. But the problem is obviously you need a lot of expertise. You need the resources to get the machine. And analysis is a whole nother beast. I'm sure some of you are experts here. But most problematic really is low throughput, right? You can only do this under kind of restrained conditions and is pretty much impossible for very, very long RNAs, or at least it's going to be very, very difficult, right? So that's the opportunity for chemical probing, right? I'm going to assume that most of the crowd is familiar with this, but I'll very quickly go through it. The main concept is you have a folded RNA, you hit it with a chemical, the chemical selectively modifies flexible sites, and then you figure out which sites are modified using reverse transcription, either stop or map, and then you put it through sequencing, right? And so because it's sequencing based, obviously this is a new development with NGS, you can get really, really high throughput things and virtually no limitation on length, right? You can, I guess, just go as high as you can, do random priming and stuff like that, and stitch them back together. It's really, really flexible. So you can, obviously, it's just a chemical. So depending on your chemical, you can do in vitro, in vivo, and in sort of really any condition you want to. And it will attack sort of different phases in your RNA, but it's a very flexible and versatile kind of method and platform that's very, very favored by RNA biologists for a reason. So this is a good opportunity to get massive amounts of data, right? But we're not quite at energetics level quite yet. So this gets to kind of the motivation behind this project. And what it is really is largely from HDX. And I'll show a slide later on HDX. It's hydrogen exchange, where they've basically figured out for proteins modeling sort of this kinetics of exchange to get energies and dynamic ensembles. So the hope and the dream is, like, can we connect this to? Can we apply some kind of HDX model to chemical probing and extract biophysics from it? So a little bit of background in HDX, right? So I guess I have this up just to show you that it's very similar to chemical probing. So there's a little H here, right? This H, if it flips open, gets exchange, right? That's KCH is this exchange. And the flipping happens at some fluctuation rate, big K, okay? And you can have it for global, so global, local, it doesn't matter in this case. But the point is, it's very similar to what we think of chemical probing, where a nucleotide opens and then reacts, right? It's very clear that the model should be able to be applied to chemical probing, and so that's what we're going to try to do. So this is like an example of a big application, I guess maybe one of the bigger ones for HDX, where they got these energies for individual residues, and amazingly, they did this for hemoglobin and correlated it with oxygen binding, right? So super direct correlation to biological function of a protein, which was amazing. And since then, obviously, it's been decades. There's like way more things that's been done with HDX, folding pathways, dynamics, intrinsically disordered proteins, and stuff like that. This is all like opportunities, right? If we can make this link to chemical probing, then we might be able to get all of this kind of amazing kind of biophysics from chemical probing. So this is the next part in the talk is this chemical kinetics view, and this talks about kind of the model itself. And before I dive in, here are some kind of additional reasons if you're still not sold on why we want to do this. At the end of the day, we still have a limited understanding of what reactivity actually is. So if you look at papers, if you've read papers, if you've seen these bar plots, if you look at the y-axis, they vary quite a bit paper to paper, right? Some papers might report 0 to 1. That's a pretty common one. Some older ones might be like go up to 15, which is this one from our lab, 10 or even 2, right? If you take a bar from one paper and you want to try to compare it somewhere else, you're not likely not going to be able to make sense of it, right? Because at the end of the day, it's the way it's treated right now, it's relative quantities, and you do these ad hoc normalization, and there's like 3 or 4 different methods to do this. So what that leaves is this opportunity for all the data to talk to each other, right? Like so many people are doing this, like hundreds of labs, I'm sure, worldwide are doing chemical probing. RMDB has like 4.5 million as of this morning. It would be nice if you could find a way to kind of allow them to talk to each other in absolute values, right? So meaning y-axis is not relative. Other point is that these reactivities are generally used to distinguish base pairing states. So obviously there are new things right now that kind of use it to resolve tertiary stuff as well. But for the most part, it's applied and used, and the way it's interpreted is if you're high reactivity, you're maybe unpaired or paired, right? What about all the intermediate stuff? There's like 12 possible sort of RNA interactions, right? There's 3 phases, there's cis and trans. Um, can we try to pull that out of chemical probing data? And I think there has been some papers that try to do it, but we want to try to do it in this more principled manner of having these exact units. So this is the main model, and we're calling it PRIME, Probing Resolved Inference of Molecular Energetics. It's really a framework. We don't own this equation. This equation's been derived at some point in the past. We rederived it clearly in the paper, so you're more than welcome to check and see it. But really, it's the framework of applying it and validating it and things like that. I just needed something to kind of refer to it. And that's what we're calling prime. So from the very base, you've got a nucleotide that flips open and closed, right? So this flipping. Can everyone see my mouse? I don't know if should I maybe use
Yeah, we
this.
can see your mouse. You can
Okay.
use this as well, but
Okay.
yeah, we can see.
Yeah, so nucleotide flips in and out like that. And so this flipping in and out really is flipping between a reactive state or an unreactive state, right? So that's open or closed. And if it's in, then it reacts with this probe. Okay, it could be DMS, could be shape, at some rate kAd. That's adduct formation rate, right? So that's the chemistry that happens if it's fully open. What's unique with shape and DMS and probing that's not necessarily as big a factor in HDX is this degradation. So we all know shape self-quenches. That's what this is, right? So it reacts with the water to degrade into an unreactive species. So there's 2 kind of competing reactions happening with the probe, and then there's an underlying fluctuation, big K, right? So depending on how much you're open, you react at this chemical rate, kAd and kdeg. If you integrate all of that, you end up with this equation. And a lot of people get scared by equations. It's really not that complicated. On the left side, that's your data. F mod is fraction modified reactivity. And this is a 1 minus exponent. Really, it's just a first order kind of rate. And so you have this fraction open, k over k plus 1 is a fraction, and you have this rate that's adduct over degradation. This looks a little funky, but this is really a first order decay because your probe goes away over time, right? So that's a very simple first order decay. And again, the derivations somewhere in supplemental, and you can go check it out. So that's the equation, right? And now we have this equation and system of kinetic equations to connect to reactivity data. This is more details that I probably already just mentioned. The main thing is this pre-equilibrium assumption approximation. So basically this flipping is way faster than the chemistry. That's why we can just call it a K. The test that I will not show data for, but I have done is basically that all of my experiments are concentration dependent. Okay, so in HDX, there's two regimes, EX1 and EX2. EX1 is kind of the regime where you're no longer concentration dependent, and so then it's actually you're measuring K open directly or something like that. We're solidly in EX2, where this flipping is so fast that it acts as a K, a fraction open, and then rate happens kind of slower. And this makes sense because chemical reactivity is going to be slower than sort of HDX things. And relatively speaking, a nucleotide flipping would be quite fast. And so that's all I'm going to say here, I think. Reiteration of this equation that will pop up quite a bit. The main highlight here is our goal is to get this green intrinsic fluctuation. And you've got all the other things here, which are sort of chemical rates, kinetic rates of the probe, as well as experimental variation, right? So there's also reaction time here. So if you pipette wrong, pipetting error would be in S, for example, or T. And all of this is encapsulated in R, which is F mod earlier. It's fraction or reactivity, right? And that's kind of what this is the data that we're dealing with today is like all of this sort of potential biases and variables are all kind of packed in. Okay. And the goal of this framework is to pull them apart and grab the K and try to analyze that and see if that's a real energy or not. Okay. So same thing that I've been already saying. I think I've been explaining the slides already quite well, but a different visualization. And it really, there's chemistry, there's Cameron variable, and we want the green K. So this is a visualization that I made a while back just to kind of picture why we're doing this and where do we stand in the grand scheme of all of the new techniques and methods with shape. We're not reinventing the experiment, right? We're looking at the data and going really deep, right? So the Earth kind of layer is where the data is. And we go deeper. One layer would be your Illumina, your gel, or your CE. You go another deep layer would be RT. Are you using RT stop or RT map, or which RT are you using? And then this light yellow and light orange really is where prime is hitting at, is this chemistry layer. So assuming that your other layers are all controlled in the same, can we validate the model and pull and separate out these two layers until we get just a yellow ball? Okay. So this is very, very deep inside any data. Okay. So a lot of the analysis that came out more recently, things like ensemble deconvolution, DANCE, DREAM, Draco, trans in-cell probing, transcriptome-wide AI training, pseudo-free energy, right? These are more older ones. All of those really are not they're affected in a different way then we're not saying that we're replacing all of that. We're saying that the data that these things use could be extracted to be more meaningful. Okay. And that's kind of the goal and vision of this project. Okay. So I'm going to move on to validation. Okay. So the key experiment here is time course probing. So this is not a kinetic reaction. I know that there have been like these time resolved studies. This is not that. This is an RNA fully folded, right, in equilibrium, just a hairpin. And then as it's reacting, you keep quenching. So I don't know if I mentioned, I am doing DMS, so it's a lot slower. I've optimized it to be quite, I mean, I think I'm using 0.15%, like much lower than the normal concentration to control pH and to make sure that I get nice kind of time courses. So this is an example, right? So you got these data points from one big tube, you stop, 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 and then you prep for sequencing and whatnot. And from the data, you can then fit it to this equation. So this equation obviously has this T here that becomes my x-axis and R being my y. And then this is a nonlinear fit, okay? So on the right is example data for a hairpin. So this is a 4U thermometer. I've got all the A sites plotted and some of them labeled. And you could see that all of them fit very, very well. And I have a guidance slide where the scores are all like above 0.8, 0.9-ish. They're all very good, regardless of are you base paired or not, right? So at the lowest end is this C25, which, oh, this guy. And that's at way under 1%, right? And it still has these nice fitting curves. And so this is our first kind of test, like, is the equation form even right? And it's an easy yes for this experiment. So the next thing we do is temperature dependence. And the reason is obviously in this one equation, there's many different rates with distinct temperature dependencies. And the goal, like I said, is to split all of these apart. And so to do that, what we're doing is this temperature dependence. So I do this time course and I do it at from 10 all the way to 85. Okay. So 85 are the really hot experiments that are just finished in 1 minute, and 10 are the ones that took 3 days, for example, right? And so I do that for the same model system, this for you hairpin. And I have some example data here just to show you what it looks like. So the x-axis are kind of in log scale time, just so it fits nicely because the high temperature ones are really fast. And on the y-axis is just mod rate, reactivity. Okay. And what you'll see obviously is that these trends might look a little weird. So the lightest color is 80 degrees. That's the highest one. And so those two will be matching between these two, and I'll show more later. But A18 being unstructured at 18 degrees is much higher in reactivity, right? So there's already something not so intuitive happening here, which is why is it so much more reactive at 18 than it is at 80? And that's explained by this equation, okay? And that's because K add and K degradation has like they're not parallel, right, with temperature. So some of like degradation probably overtakes add at high temperatures, which is why at high temperatures it's much slower. So there's like things like this that already are starting to pop up that we're gonna hopefully formalize a little better later. But I like this plot just to showcase that like we don't really know fully how to predict reactivities naively, right? So then we dig into degradation rate. So this again is the rate at which DMS reacts with water or buffer. So I measure this rate independently using NMR. So it's literally DMS in my matching buffer, drop it in 1D proton, and just track. So I thankfully I know these assignments because there was already a paper on it. And so I could very clearly see this DMS dropping, and then you got MMS, so monomethyl sulfate going up, methanol, and then there's like some other products that interacting with my buffer, very small amounts. But generally speaking, what we care about is this DMS dropping, right? So I do this experiment at 5 different temperatures. At least the higher temperatures I can't do is too fast for NMR because I need to like rush to put the tubes in and stuff like that. But I was able to get these rates independently, right? No RNA. And then at the same time, for the RNA side, what I did was to do a global fit of all the sites. So I have, I don't know however many sites here. During nonlinear fits, each one of them has a curve. And all I'm doing is I'm saying you all have the same degradation rate. So I have another figure that I'm not showing why this is valid. The main reason is inside your tube, most of the DMS or probe is reacting with water because of the concentration excess, right? So normally we do something like 10 millimolar or more of the probe to 1 micromolar of the RNA. So this is a very valid thing to do. And I have some figures to show that it actually helps with fitting because there's too many variables that are shifting around. So it helps to lock it into these global things as a constraint. So anyway, when I do that, and I plot probing extracted rates versus independent rates, that's what this plot is, right? And this was a very impressive result. I was very happy to see this. Red dots are prime, so that's experimental probing. And then black is NMR, direct hydrogen tracking. And this is incredible because this is probing, right? It goes through RT sequencing, all the pipetting errors at high temperatures, at low temperatures, and the line basically is on top of each other, right? And this was an amazing result. This is for one of the rates, degradation rate. And on its own is already very cool because you could potentially apply this model to cells and get degradation rate in cells, right? Where NMR is not even possible or at least very hard. So this was the first kind of like fascinating slash exciting result from this model. I mean, obviously we had it fits well to the equation, and this was really the first one that said, okay, we're onto something. So the next challenge is kAd, and this is a lot more complicated than it seems. So kAd is the adduction rate, so reaction of DMS with a base or a nucleotide. This is combined in a composite rate with big K. So how do you split apart two rates, right? Because when we do these fits, you only get K observed, and you want to try to get one number out of it. So again, this is why the temperature helps, because the idea is at high temperatures, you melt the RNA open. And when the RNA is fully melted open, your K approaches infinity, so like very, very open, which means the K over K plus 1 approaches 1. Okay. So that's how we get rid of the K effectively and get kAd. So this is all in theory based on the equation, right? So the definition being kAd is the maximum possible reactivity. So because K over K plus 1 has to be under 1. That's a fraction, always going to be under 1. And so kAd has to be the maximum possible rate, which we achieve by melting. Okay. So then the hypothesis would be if I had these rates for each of these sites and I melt it open, they should all converge, right? And that is actually what we see. So I'm going to direct your attention to the middle because that's what I'm talking about right now. So this is all log K observe for all A sites. I also have this for C sites in the paper. I'm just showing the A's. Low temperature 10, and as you go up, they all kind of converge into a tight kind of squeeze up here, okay? I've got standard deviation as well, showing you kind of the same thing, where as you go up in temperature, they all melt and agree, kind of like match to each other. They're limited really by the chemistry of an A reacting with DMS, a fully open A, right? And so that's the kind of like convergence that supports kind of the hypothesis I just talked about earlier. So once you have that, right, this is log, you can plot it on an Arrhenius plot, and you could see that it's actually, quite linear. Obviously, this is very high temperatures. They're like 65 to 80, higher error, but even then, it's still linear. And more impressive is I did the same independent experiment using NMR. So, in my NMR tube is literally DMS buffer and ATP. So this is ATP. And I do ODE to match kind of like how the products change, and I can talk more about that. But it was really, really cool to see them line up perfectly on the same plot, right? So same thing. The black stuff is directly from NMR. They're tracking hydrogen changing and sort of shifting. And the red is not just time course, but this entire kind of extrapolation down at high temperatures and sort of convergent over multiple sites. So this was another really, really, really exciting result to see that now the second rate, there's only two rates, like degradation and adduct, and now this adduct is also validated. So that's what we got, right? So adduct is validated, and this is a reminder again on what these things are. So add is NTP plus probe, and then the other one's probe plus buffer or water. And so really we're set to extract big K, right? This KNT. That's what we're going to do next. But before I show you some data, I wanted to clarify again sort of what this K is, because I think this will show up again maybe for questions later. This K is fundamentally flipping between a reactive and unreactive state. These two states are coarse-grained, right? So they are not stacking, they're not base pairing, right? They're kind of gated by what is reactive to DMS and what is not. So that's an important distinction to make, is because there's a lot of within reactive and within unreactive, there's probably a lot of RNA motions that are captured. This is coarse graining, and in some ways it's kind of an artificial kind of cutoff, right? Because of the coarse graining, where reactive is basically anything that DMS can touch, right? So it opens right just enough for DMS to touch, for example. So so I guess to just set some numbers in your head on what the delta G's should be around. For base pairing, like I said in the first slide, it's it's debated 1 to 3 kilocalories per mole or 4 to 7 or something like that. This is from Hashim's and Niels Walter's kind of review of the span of different motions. We're going to be somewhere in between the two. Okay, so that's the energies that we're seeing. And if we see energies in those regions, then that means we're capturing actually these types of motions and stuff like that. But at the end of the day, it's going to be probe specific, right? So if you are doing shape, that's a very different set of sterics and electrostatics that you need to think about, which means your delta G might be different. Okay. So this is the data. So this is 4U thermometer. And I have two variants, wild type and AHC. I forget to put the citation. So these are mutants that were measured by immunoprotein exchange by the Schwalbe lab. And that's why we measured it, so that we can compare energies later on. But for now, we're just going to look at sort of my data and kind of see what new things have we learned from this. First of all, right, this y-axis is no longer an arbitrary rate. It is now actually in kilocalories per mole. So you go from 0 to 4, and like, it's giving a lot of like math equations, right? So you go from K observed, now you have K add, K deg, you extract all of that, you go to K, and then you get delta Gs. And this is basically the range, right? So 0 to 4 kilocalories per mole is actually physiologically relevant, right? So going back to this, it's like in solidly here, right? Again, we're not doing librations, I don't think, or maybe it is, but it is definitely within physiological sort of like range of energies that we're capturing. And so the key sort of sites here are this mutation. So AHC mutation changes an AG to a CG. So naturally, what you would think is nearest neighbor rules. The nearest neighbors will be affected, which is what you see here, right? So 7 and 9, its neighbors are much more stronger in the mutant, and then everything else is kind of less. But the more interesting thing that I am very convinced is real is this A18 is changed. Okay, so this A18 is all the way up in the loop. That's supposed to be unpaired, right? In maybe in nearest neighbor terms, it's just one of the tetraloops. It's not even a known tetraloop. But there is I think that's a real energy change. It's small, but I think it is real. And I'm convinced that there is possibly some kind of propagation in energy that goes from this bottom strand all the way to the top. And I'll show more examples of that later. But these are kind of some of the early things that we're seeing. So the other thing that I want to note is the difference between an AU and a CG base pair. Okay, so an AU would be I wish I had it colored better, but it's on the range of 2 to 3-ish, or maybe a little lower, like 1 to 3, 1 to 2-ish. And then the CG, there's only two C, so there's the C25 and C29. They're closer to 3 to 4. Okay, so these are higher, but there's a distinct kind of like higher for CG and AU, which makes sense, right? You got 3 hydrogen bonds versus 2. So I'm giving you kind of a sense of this is like our classic model system of a simple hairpin. It should be a regular helix, right? There's no funny business here. Maybe the AG is a non-canonical, but largely, especially the CG, should be a regular helix. And so you would expect that they would compare well to sort of nearest neighbor, for example. And that's what we see. So let me break this down. So there are two reference energies that we're comparing to. The first is NMR. So this is an experimental measurement, again, by the Schwalbe lab, so this paper, where they directly measure base pair opening via G's and U's. Okay, so I'm measuring A's and C's. They're looking at G's and U's. And how it works is there's this hydrogen donor that comes, so this base comes in and grabs this H, and then it exchanges with water and so on. And this is measured by NMR. So this is a direct sort of hydrogen sort of energy measurement of this energy. And the idea is if the base is open, then it will exchange, right? Very similar to kind of how DMS is thought of. And that was a really good reference, right? And for that, we have this very nice solid R square of 0.8. And then the other one is nearest neighbor. So it's a little tricky because nearest neighbor is in quartets. Okay, so it's in base pair over base pair is kind of how it's calculated. So Dave Matthews, who's an author on this paper, helped us come up with this scheme where you basically force a base pair to be open at each site, and then you get the penalty of opening. And so that's what this correlation is you calculate the penalty at each site, and then you correlate that against the energies we get from DMS. And once again, this R square is really incredible, right? Like R square of 0.86. And again, I think this all checks out primarily because of how regular this helix is, a very small helix, very canonical. And so yeah, so this is like a validation, very early validation on the case being actually physiologically kind of real, right? Or at least it seems significant. I have an additional plot here just to show that NMR and nearest neighbor is actually not perfectly correlated, which means we're capturing different features from each one. So I will note that correlation aside, these are all R squares, right? We could have settled with just, R squared great, but we also notice the scale is off. Okay, so my scale goes from 0 to 4, that's DMS. NMR goes from 2.5 to 10. Okay, so the slope is around zero point 0.5. It's like half. And the same thing for DMS, right? So, sorry, for nearest neighbor, same thing, 0 to 4, and here's like 0 to 7-ish, okay? Which is still kind of off. And there's like questions of why. Why is there a disparity in this magnitude? And this gets at the title, right? Like this ubiquitous low energy thing, which is kind of alluding to. This open state could happen in many different ways. Okay, so I have like these are absolutely not to scale. I just drew this for visualization, where maybe a low kcal shift is something very slight, like a small open like that. And a bigger one would be a full extra helical extrusion. Okay. And so what I'm suggesting here is maybe this disparity in magnitude is due to the other methods requiring this full opening of the base pair, whereas DMS is capturing this smaller kind of breathing almost things at 1 to 2 and maybe 2 to 3. And this makes sense for the nearest neighbor naturally, because the nearest neighbor, you actually force both out. And the nearest neighbor parameters are they're calculated from optical melts. You melt the entire thing. So yeah, so then it kind of makes sense for nearest neighbor. Like there's some redundancies, you're calculating stacking more than once and stuff like that. But for immunoprotein exchange, this is a little unclear. So we've spoken to Hashimi about this and their lab, and they also suspect something might be happening where maybe there is an intermediate state where it needs to open before the base could come in. So there's a structure dependent aspect to it that's artificially increasing the energy, right? So like I mentioned in the very start, the original tritium exchange was at our range, so like 1 to 4, right? But then the new NMRs, they're at 4 to 7-ish. So they're like much larger energies. And so this is not fully resolved and probably an active kind of area of research as well, is whether or not the NMR immunoprotein exchange is actually measuring more energy because of some kind of extra step. Okay. And again, I can't comment more on this, but this is kind of our observations and interpretations and what we know. And or we're also kind of building an intuition on how we think about these lower energies. So we think of it as these small kind of breathing, almost kind of motions. Okay, so what about more complex RNAs? So earlier, right, like I said, like we post vanilla kind of duplex, simple, short, and stuff like that. Now that we've kind of validated it, what does it look like for more complex things? And what do we mean by complex? There's two things. One is HIV tar, and two is P4P6. So I cite the RNA as CASP SIG talks because I think they're fantastic. These two people know way more than I do for these methods. And they can probably give you the background that you're looking for. But I'm primarily focusing on these systems because they are much more complicated in interactions. For HIV, it's because it forms these dynamic ensembles that's been rigorously measured with NMR, R1 row from Hashim's lab. And for P4P6, it's all these non-canonicals, right? And so, yeah, so I would totally check out their talks. I checked them out before I made mine. And I think they did a great job explaining in a lot more detail, all of the sort of background behind this. For Joe's, it's not specifically P4P6, it's actually this TL-TLR interaction. But regardless, these are significantly more complicated systems that we wanted to look at their energies at. And for P4, P6 specifically, if it's true that we can get biophysical information, if we can prove that it works with probing for a large system like this, that would be very significant, right? Because you can basically get biophysical information for as long an RNA as you want and as complicated as you want. But little did I know this is like a lot more complicated than it is. So the parts of the analysis here are kind of like how we think about things based on the data that we got. And obviously, there's no real reference for these things because as far as I know, there's no measurements for some of the data that we have. There's no reference data. We can only infer from what people have known about these model systems. So I'm going to start with the wild type TAR. Okay, so TAR exists in three states: GS, ES1, and ES2. They have more states that they're defining now, and this might be outdated, but generally speaking, these were from their earlier papers, even maybe 2023, where it's 87 most of the time it's in this GS state, and every now and then 10% of the time it's in ES1. So the difference is this A35. Okay, so this A35 and the CG, so the CG and the A35 kind of swaps with each other. So this A35 flips in and interacts with C30, or it goes with the G. So there's this like flipping that's happening dynamically that they see in NMR. And then ES2 is also very interesting. There were maybe two or three papers just on this because it's so low percentage, right? So it's 0.4% based on their measurements with NMR. And what happens is the trinucleotide bulge, the orange part, base pairs with the other side with a lot of these non-canonicals, and it shifts. It leads to a register shift of the loop into this elongated helix. Okay, so this is supposed to be basically impossible to see. All right, and we were determined to try to find it, and hopefully you'll see some evidence that this is true. So wild type, I have the bar plots here. So the color bars and the bar plot are all kind of matched. The first thing to look at is canonical base pairs. Like, what do they look like? And so it's really mainly the sites in SL1, SL2. These are stem loop 1, stem loop 2, and they are within 1 to 3 kilocals per mole. All right. So same consistent thing with 4U thermometer. We're under 4 and closer to 2-ish to 4. C's are higher, right? So C37, C39, they're closer to 3-ish, whereas the A's are weaker. And then A22 is at the edge of this trinucleotide loop. So, it's kind of, I'm blanking on the term, but it's basically more accessible because it's getting pulled out occasionally or way more than A20. And then C24, this is the first time we're seeing a negative delta G. So C24 is the trinucleotide bulge, which is a very characteristic feature of TAR, and that's flipped open. Okay, so a negative delta G just means it's extremely highly reactive. So this all checks out. And then the last thing I will mention is this A35, right? So this A35 is the one that's flipping in and out. And what you see is actually an intermediate delta G. So it's lower than a comparative kind of canonical A, but it's higher than the negative C, right? So it's kind of in the middle, which makes perfect sense for this type of model where it's kind of flipping in and out, okay? So obviously we're not going to stop with just wild type. We're going to look at some mutants. And I'm just going to point out certain things. It was a little hard to visualize this, and I hope this is not too confusing. And what these mutants do is they clamp the top. So I have the little cartoon here to show where the mutants are. And the mutants are all kind of like clamping at this loop region, okay? And all of them are changing something into a canonical. So it used to be an AC here, and now it's sorry, CA, now it's a AU, and a CG here. And what we want to observe is how does it affect reactivities or sort of energy in the others? So C30U, I think, is among the more interesting ones because we have direct evidence of this A35 being much. So these are all delta delta G's relative to wild type. So A35 is definitely more stable. That makes sense. The C24 is actually it's not as negative anymore. So it became more constrained, right, with this C30U. In A35G, it still is quite negative, and UUCG, another mutant, is also quite negative. But the C30U is particularly interesting because what that means is this 24C is constrained in some way that we don't know. And this is invisible to NMR, by the way, because anything that's open and flipping too fast, they don't really see it. What they can capture are solid base pairs, right? And so we're really capturing information that they haven't seen before. And maybe they have suspicions on models and why this is, but this is a good example of this C30U might be sampling, ES2 a lot more than we think. But the point holds that the TB, so the C24 site, varies quite a bit across these mutants. In particular, this ES2 is very obvious, right? So it's the C24. And we did one stop, I guess like we didn't go all the way to extracting populations, but we have a path from prime to do that with these things, maybe with more mutants that we can more confirm, like more confidently assign sort of mutations. But yeah, so this TB are all variations in flexibility that came as a result of strengthening the top loop, right? And so this is what we're saying is a distal energetic coupling where you strengthen something at the top and it affects things at the bottom, right? We do some more interpretation in the paper on some of the loops, so some of the stem loops as well. But hopefully that's sufficient to show you sort of how with energies, we're actually very, very sensitive to context, right? If you change something beyond nearest neighbor, right, you actually still see it. And I already showed an example with 18A from 4U, and we're seeing it again with HIV. So this last part is going to be on P4, P6, and this is a bit of a monster beast. So I'm going to keep it brief as much as I can without losing the audience. So what we did was we did the same time courses at 23. So we matched Dan Hirschlag's conditions at 5 millimolar magnesium and also at 0 millimolar magnesium. So what I'm showing here is at 5. So this is when P4, P6 folded as it is. This is annotated as it is on the 3D structure, the crystal structure. So you'll see some of the non-canonicals all annotated, and that's all based on the crystal structure. So the first thing I want to say is this non-canonical, and I want to drill this in again because it's significant. So you filter out all of the non-canonicals and group them, and these are delta Gs in the presence of magnesium. Once again, you're in this range of 1 to 3. Okay, so we see that again. We have some sites here that we don't have data for because it was too low. Okay, so that's greater than 3. Those are much tighter interactions, and those are typically the CGs, and you'll see them as black on the structure. But once again, 1 to 3. Similar range as HIV, similar range as 4U. You look at non-canonical. So NC is non-canonical local. So what local means is it's not a long distance interaction. So it's a Hoogsteen sugar or a Hoogsteen Watson-Crick or something like that. And those actually are lower. So they're like 0 to 1. And the reason is because some of these are flipped out, right? So if you're interacting with Hoogsteen, your Watson-Crick phase will be accessible. So that's kind of where that is. And I'm lumping that there and I'm not thinking about it too much more. Maybe I'll mention it again later. But then there's these interesting kind of tertiary interactions. So that's the sites that are kind of linchpin sites known to hold this whole thing together. So you have the P5C metal core, and then you've got the TLTLR, and then you've got also a junction. And those things and those sites are actually in kind of a comparable region to canonical. They're not necessarily much stronger, right? They're actually in the same kind of region. And later on, I'll have a clarifying slide on why we can make this comparison. But for now, this dotted line is our reference energy. This is a global delta G fold from SMFret. We took this value, this 2 kilocals per mole from their SMFret data from Anne Hirschlag's lab. And the idea being, if your entire thing opens and closes at 2 kilocals per mole, your local should stay below that, right? Because when it opens, then the local kind of fluctuation should be just free and open. And so that's kind of what we're seeing a little bit here is it's actually gated, right? So it's gated by the global energy value, which is kind of exciting because we're comparing completely independent methods and scales and making sense of it with our values. So how do we explain these things on above, right? So the thinking between for the things above is things that are kind of within the folding pathway is either more packed or requires additional steps to unfold or things like that that we didn't explore too much more deeply beyond that. But we do see them, right? So these are in the range of like three and greater than three. They are comparable to the strongest CG pairs. So that's another thing that we can say about that. Unpaired, also 0 and 1, nothing too remarkable there, consistent with 4U and HIV. This is the delta delta G. So delta delta G is comparing 5 millimolar magnesium from 0. So no magnesium, meaning it's just secondary structures. Um, and the key thing to note here is the canonical base pairs are mostly above 0. So, obviously tertiary will change a lot. Tertiary interactions will strengthen quite a bit. But the canonical are actually slightly above as well. So what that means is the tertiary interaction, the formation of these contacts, is actually tightening up canonical a little bit as well, right? And so that was really interesting. And I think I have another slide to talk about that a little more, but I'm running out of time, so I'm gonna try to keep it really brief. This one I wanted to talk about because Joe talked about this in his talk of supporting this model from Julie Fagon for this TL-TLR interaction. The finding we found is there's an asymmetry in energies. So the light blue is all tetraloop. They're all around 1. And then on the other side is the tetraloop receptor, which are ranges from 2.8 in the strongest one to 1.9 and zero point 0.7 for some of these that are more kind of sugar interacting. And the thinking is that this TL darker region is pre-forming in some way. They're naturally already a little more stable. And then the TL comes in kind of in an opportune position. And so then they form into the pocket, but they can still fluctuate out in and out independently still. And so this value of 1 is on the weaker side of a canonical base pair, right? And so they would be, I would say, susceptible to base pairing. They're not really strong in as they are kind of locked in. And so this is why we say it's supporting this NMR TLTLR from Julie Fagon's lab because of this asymmetry that we're observing. I think I'm going to skip this in the interest of time and skip to this last figure. So the title of this results section is secondary tertiary energetic coupling. And this gets back to the point I mentioned earlier, which is why are the canonical interactions like above 0 when they interact? The previous thinking is they're perfectly modular. The canonical regions are just links between contacts. And so the thinking that we have now is, primarily because no one's ever measured it before, no one really knows, and now we have access to this, that tertiary contacts has actually the capability to strengthen the canonical pairs. And for the first time, we actually have a way to quantify them in terms of kilocals per mole. So this is a rudimentary analysis where we summed the energies to look at relative contributions. Again, there's a lot of caveats, right? But this was really interesting to look at because you could see that the addition of magnesium leads to 34% of the net increase in local stability for canonicals. Obviously, it's dominated by tertiary, that's expected, but the canonicals is also very, very interesting. So as an overall kind of molecular model, so you have all these interactions stabilizing it, and then you must have some kind of energy for conformational entropy that's lost, right? And that number from literature is estimated from, I think, Dan Hirschlag's like MD simulations or something like that to be around 10. And that's kind of what we're not saying anything concrete here, really just showcasing the type of analysis that could be done. Obviously, these are very rudimentary, right? There's a lot of assumptions, but we could come up with models like this to study electrostatics and other things like that to more quantitatively think about energies of non-canonicals relative to canonicals, right? Which we can do now for the first time. Okay, so this is wrapping up. I really wanted to clarify this state that we're measuring. They are reactive versus unreactive, and not, stacking or some specific thing. Underneath might be a bunch of different states, but it's a coarse graining of reactive, unreactive. And so at the end of the day, this is going to be probe dependent. And the other thing I want to point out is the fact that we're measuring intrinsic fluctuation. This is independent of any addition of ligand, ion, metal. Like, you can measure this for anything, anything that has a reactivity, basically. And this is different from a delta G that you might be used to, where, for example, for SM-FRET, you might be titrating magnesium or titrating ligand. Those are all conformational delta Gs, right? Because you're measuring kind of how much of the conformation has shifted. In our case, so I guess that would be meaningless if, for example, one of the sites stays unchanged throughout the time. In our case, we actually have a measurement, and we can actually measure the intrinsic fluctuation of that. And the reason is our criteria for states is just reactivity, and anything can react, or at least anything has some kind of reactivity that falls between this fully reactive and unreactive. And so that's kind of how we think about this delta G. And I really need to clarify that just in case it's not clear. So final conclusions, we are now able to decouple the chemistry from intrinsic biophysics with the model. Like nucleotides universally undergo this lower energy fluctuation than previously thought, which means they're actually a lot more accessible than we think they are. Energetics are coupled and distributed out context dependent, not strictly localized. And the final thing is kind of on an application where Prime can really open up kind of the floodgates for scalable biophysics at nucleotide resolution. The last thing here, this just came out recently from Hashim's lab. It is one of the cases of, I guess, showcase on what you could do if you have these energies very well done. So they have this for tar, which they connected directly to binding delta G and then to cellular activities. This is kind of the dream, and they've been restricted to HIV and to relatively small. This is probably the largest library they've done. But for probing, you all know we can do this like thousands, 10,000s, millions more, right? And so this is really an opportunity for the entire field to hop on and start analyzing data in terms of these energies and connecting them to function. Okay, this is my last slide. Next phase, clearly this is a very foundational type of project model, and there's a million different next steps to do. These are the three in my mind that are kind of general directions. One is standardization. How do we make future experiments more easily, I guess, like allow data to talk to each other better, right? So the goal is to change all our y-axis to these kcals per mole rather than stick to arbitrary reactivities. The second one here is measure. So there's a lot more things to measure: protein binding, dynamic ensembles, and stuff like that. Motifs, 3D motifs. What are those energies of formation, stuff like that. And then there's develop. There's a lot more theory to develop. There's like EX1, fast, slow regimes, microscopic to global. We did a little bit of that, but not rigorously enough. And so there's a lot more work to be done there. So a lot of this really is on decades of HDX work, right? That we're kind of trying to track and see how we can accelerate the RNA up to that speed. And so the hope and dream is this, is accessible enough for everyone to be sold on it and start building on top of it. We're not looking to replace anything that's out there, right? We're kind of encouraging people to rethink about what your data might actually have. You might have more than you think. So with that, I'm going to end with the acknowledgments with my lab, obviously. So Julius, Cade has been fantastic, helping me out with a lot of the probing experiments, but really a lot of discussions with everyone who's in the lab. Tony has been a very, very, very good collaborator for many years throughout this entire project, not just on map protocol, but in a lot of the energetic thinking as well. Dave came in as well a little bit later, but on nearest neighbor thinking and also on energetics. And also lots of very good discussions with various people: Gabe Rocklin, Dan, Joe, Riju, Hashim, John, and Yongjun. And I'm sure there are more that I've spoken at poster sessions that contributed to this that I might be missing. With that, I am happy to take questions.